And welcome back, everyone. I'm Anna Berry, happy to continue on this fantastic conference today. Next up, we have Dyadic International, Inc., trades on the NASDAQ under the symbol DYAI, and is a global biotechnology company focused on improving and leveraging the patented and proprietary C1 expression system to help bring biologic vaccines and drugs to market faster, in greater volumes, at lower costs, and with new properties to drug developers and manufacturers to improve access and cost to patients and the healthcare system, but most importantly, to save lives. Today, we have President and CEO of Dyadic International with us, Mark Emalfarb. Mark, it's great to see you. Welcome.
Thank you. And that was a great introduction because it really talks about our platform. And it's a platform technology that is broad and deep and can do thousands of potential good things for humankind all over the planet. So we're going to get into that, and we appreciate your introduction. So we're very pleased with the progress we made in 2021 already. And in 2020 was a year of considerable disruption for the world, with suffering and death worldwide as COVID rapidly spread across the globe. We feel that we have a better, more efficient way to develop vaccines and drugs faster, quicker, and at lower cost, at larger scale that can be produced globally and regionally. So here we go. This is our safe harbor statement. So as you can see from this slide, Dyadic at a glance, we have a very strong financial position. We just last night reported our year-end numbers. So we at 12/31 had $29.2 million in cash, no debt, no warrants, and a revolutionary technology that we're applying to human health, animal health, infectious disease, metabolites, growth factors. So really what we're doing here at Dyadic is we've spent over 25 years engineering this hyper-productive way to make vaccines and proteins in a large scale, very low cost on the industrial side. We use it to make enzymes for feed and fuel and biofuels, sold products in 35 countries around the world ourselves, and licensed it to small companies like BASF, Shell Oil, and others before DuPont purchased the industrial biotech business for $75 million on 12/31/2015. Since then, we've been re-engineering these cells to produce vaccines and drugs quicker, faster, and cheaper. And so some of the things that we're doing here is we're building a more efficient way to produce biologics. Biologics are things like insulin, cancer drugs like Keytruda, rheumatoid arthritis drugs like Humira, Enbrel, EPO. So it's really an endless opportunity for us and many shots on goals, because what we're doing is we're taking gene sequences from our collaborators in the animal health and human health business and in infectious disease and governments and academics. And we're taking those gene sequences and putting them into our cell line, and that produces the protein. And the protein can be a cure for cancer, it can be a treatment, it can be a vaccine, it can be a biologic, it can be a nutritional supplement. So all kinds of opportunities. And we've spent over the last 25 years between us and Shell and Abengoa and BASF and others, Codexis, the way to program these cells so that we can take these gene sequences, put them into our cell line, and mass produce the products that the world needs in larger amounts at lower cost at flexible commercial scales. And we have all the genetic tools to do that. You've heard of things like CRISPR. We have our own CRISPR-like ways of engineering these cells rapidly to produce large amounts of low-cost proteins. And this can be used all over the world for a lot of different things, and we'll get into that. So it's a very versatile platform. It's robust. We can use it to grow things at massive scale if you want. We used to run at 500,000 liter scale. And in the pharmaceutical world, the largest tanks are around 20,000 liters. So we've run at 25 times larger scale. So we're usually in biotech, it's failed to scale, and we're going the other way, we're scaling down. Because in the industrial side, we needed to make things for $1 a kilo, not $1,000 a kilo. So we're bringing the power of industrial biotech into the pharmaceutical industry to disrupt the way things are being done. And as you can think about, as an example of Elon Musk in the electric car business of how he had the dream and the vision, and everybody said it couldn't be done. But guess what? A lot of people driving Tesla cars, and all the car manufacturers are now making electric vehicles. And so we believe the same thing's going to happen here, is we're going to open the floodgates as soon as we get our first human trial in drug, in this case is a COVID-19 vaccine, which we'll be talking about later on. And so we've developed business developments with all four of the top animal health companies and another global animal health companies with their funding projects or have funded projects to test drive this technology to see if we can help them produce more for less for animal health. And animal health is basically broken down into farm animals, which is chickens, pigs, and cattle. And then you also have companion animals like dogs and cats. So there are two different markets and two different opportunities. And then the global strategic partnerships we have with big pharma, multiple people that we're involved with, from big pharmaceuticals, biotech, governmental agencies, and we'll get into some of that as we move into this presentation. The markets are massive. If we just get a small fraction, even 1 or 2 or 5% of this, it's a huge opportunity. But what we're doing here is we're not addressing these markets directly. We're addressing them indirectly, like the picks and shovel the gold mine. We're the providers of a more efficient way to manufacture these drugs and these vaccines. And a few of those we're taking on our own and advancing to prove that we can do this in a safe, effective way. And we'll talk about how we already have significant data that demonstrate that's the case. So what we're doing here is we're building on the success of 2020 and the momentum that we built from the pandemic, because the pandemic changed everything. It highlighted the inefficiencies of the biomanufacturing processes, the failure of big biotech and the governments to deliver vaccines in the quantities and at the cost that the world needs. So we think we have a better way to do it. We've demonstrated it, and we're on our way to prove that out on a variety of applications. Again, in animal health, farm animals, companion animals, human health, biologics, antibodies, therapeutic proteins, bispecifics. We'll get into some of those as we move forward here as well. And our management team, we'll get into it, and our board members are world class. So our focus here to start with, because the pandemic's here and it's upon us and it doesn't look like it's going away anytime soon, as the variants keep emerging, and we'll talk about how we're going to address those variants, and we think a more efficient way to actually help save lives globally using our technology. So we're advancing towards our first phase 1 clinical trial using a Dyadic-owned C1 cell line, which is called DYAI 100. We demonstrated already safety and preliminary efficacy, and now we're working on doing that as a proof of concept in humans. We've used C1 in animal studies, in cattle, in sheep, in mice, and it's been safe, it's been effective, and it's shown potency. We continue to advance our commercial recombinant antigen vaccine products in animal health with global animal health partners. We're generating safety data within the Zoonosis Anticipation Preparative Initiative, and we'll talk about that coming up. We spent 5 years producing the Schmallenberg virus antigen and also the Rift Valley fever antigen. And again, we demonstrated safety, efficacy, and potency, and in hyperproductivity at 300 times the level of the second place cell line. And we'll talk about that as well. And then on the human pharmaceutical programs, we have not only working on one of our own products, nivolumab, which is a potential biobetter or biosimilar to Bristol Myers Squibb's $8 billion Opdivo, but that's a proof of concept. So if we can demonstrate we can make a monoclonal antibody at the right quantities, at the right levels, with the right properties, there's hundreds of other molecules that could come behind that. So on slide number 9 here, we're talking about this European Union Zoonosis Anticipation Preparative Initiative, ZAPI. They just had their final stakeholders conference, and we're going to take a few slides here and talk about that. As you can see here, Dr. John Christophe Audonnet, who ran that program, called C1, far exceeded their expectations. We produced massive amounts of Schmallenberg SPV antigen and RVFV. And again, they were safe, protective, and effective. And on slide number 10, I'm going to play for you what Dr. Albert Osterhaus, Erasmus Medical Center, who's one of those scientists within ZAPI that had firsthand knowledge of experience over the last 5 years preparing for the pandemic and what would come. And here's what Professor Osterhaus had to say.
The manufacturing processes here, there's the efficient manufacturing processes are being developed. I specifically would like to mention Dyadic, the C1, the fungal process there, which gives enormous amounts of antigens. And I think, especially in the face of the world situation with the pandemic, a system where you can produce much more vaccine would be an ideal one to look into. So if that is if that were to be the only spin-off, that would already be fantastic.
So you can see Dr. Osterhaus and what he had to say about C1 is nothing else came out of those 5 years. It would be fantastic on the results that they achieved with C1. And so Dr. Osterhaus, just so you know, is one of the top 20 coronavirus scientists globally, and 2 of the other members of the ZAPI consortium that we're working with are 2 of the other 20 members. I think one's number 5, one's number 17, and one's number 18 out of top 20 coronavirus experts. This particular slide now is one more scientist. This is a scientist, Dr. Alexander Brix, who works at a commercial company called Boehringer Ingelheim. If you're not familiar with Boehringer Ingelheim, they're a big pharmaceutical company, and they're one of the top 4 animal health companies. Let's hear what he had to say about C1 and the technology and the firsthand experience that he and the other scientists had dealing with C1. Up. Sorry about that. It looks like we had the same presentation by Dr. Osterhaus. So somehow the video guy didn't get that right in the presentation. But if you go to our website, you'll be able to see and hear what he had to say in that presentation as well. So on slide number 11, let's see if I can get it to work now. So hold on. Nope. They put the same text in for both. But as you can see here on slide number 11, Dr. Brix had to say they selected the C1 technology. They selected the C1 technology, and you can read what he had to say here. But basically what he's saying is not only can we make massive amounts of protein at very low cost, but we can do it at flexible commercial scale. And what that means is you can produce it locally in small tanks. He talks about going from 4 to 5 to 6,000 liters down to 100-liter tank size to get the same output. So our technology can be tech transferred all over the globe using microbial fermenters. On slide number 12, this just again demonstrates one of the examples here is we produced the SPV antigen in that program that again in animal trials was safe, effective, and protective, but we produced it at 300 times greater productivity levels than insect cells or baculovirus cells. And to note, those insect cells and baculovirus cells are the cell lines being used by Sanofi and by Novavax for their COVID-19 and their flu programs. So we can make things at dramatically larger amounts, lower cost, at flexible commercial scales than those existing cell lines that are being used by Big Pharma and by biotech. Another collaboration we had, and again, this one started in 2018, 2 years before the pandemic, we developed a relationship with the Israel Institute for Biological Research. And the goal of that was to prepare them for biological threats, pandemics, epidemics, and bioterrorists. And initially, we produced an enzyme that worked very successful in producing with the IIBR that prevented toxin human nerve agents like sarin and VX gas. When the pandemic hit, we expanded our collaboration in February of 2020, and we worked with them to develop a C1 cell line that could produce a SARS-CoV-2 or a COVID-19 vaccine rapidly in large quantities at very low cost at flexible commercial scales, and we succeeded. And then they moved that forward into mice trials, animal studies, which were successful at demonstrating high neutralizing antibody titers, and also they could produce things in a very rapid way. They then did the studies for ACE2 transgenic mice, and that worked out in the challenge studies very good. And that led us to start, what you'll see here, our first in-human phase 1 C1 cell line produced COVID-19 vaccine that's moving towards a clinical trial. We developed that on our own in conjunction with the Israelis, with the Europeans, and that's a Dyadic own vaccine candidate. And I'll talk about that in a minute. But you can see on the right are the people like Albert Osterhaus and the other top coronavirus SARS and MERS scientists that did the animal studies, and the Israelis did those studies as well. And we're guided by Parexel, which is one of the largest consultants for regulatory and CMC consultation, and then the CGMP manufacturing for the toxicology batch, and the clinical batch is being done by Biotechnology General, which is an Israeli company. It's a biopharmaceutical company that's a subsidiary of Ferring Pharmaceutical, and the clinical trial is being overseen by CR2O, who's run numerous infectious disease trials. And the goal here of this trial is to prove, first and foremost, safety and preliminary efficacy in human beings. And then from that, we can then reprogram, which we've already started, these C1 cells with the Brazilian variant, the UK variant, the South African variant, and use those technologies to create what's called a trivalent or a tetravalent vaccine that would be 3 or 4 antigens produced in the same vaccine candidate, just like influenza. You may or may not realize, but when you get a flu shot, it contains 4 different proteins. And if the world's having trouble today making 1 protein, I'm not sure how they're going to make 4 cost effectively without a technology like our C1 technology. And our strategy here is to make things for a global population in large quantities affordably. And we can program our cells very quickly with the new variants that are emerging. So on an annual basis, if this COVID-19 pandemic sticks around, we're there to produce more for less at a flexible scale regionally around the globe with our partners. And we just announced recently that we made a deal and a partnership with a company called Meditox Inc. It's a Korean company. That Korean company is the 4th largest producer of Botox in the world, and they're working with us on COVID-19 viral variants that are emerging all over the world. And then we're also working on antibodies. We have produced a successful monoclonal antibody with C1, with the ID Biologics, which is a small biotech company that works with Vanderbilt University. We've executed a material transfer agreement with a large pharmaceutical company that is fully funding the research and evaluation of a C1-produced SARS-CoV-2 monoclonal antibody. And with the group in Zappy and these scientists, we're working on and expect to enter into a collaboration to produce an antibody that hopefully we can get into clinical trials towards the end of this year as well, or the beginning of next year, for an antibody for COVID. So the point here is a platform R&D technology transfer. We can transfer this technology very easily, very quickly. We've already have a non-exclusive research agreement with WuXi Biologics, which is one of the top 10 pharmaceutical contract manufacturers of biologics in the world. And we're engineering C1 and modifying it to do the several different things that we need to do. So when we moved over from industrial biotech, we had this really powerful industrial workhorse that could produce massive amounts of proteins at very, very large scale at very, very low cost. We had to re-engineer it and remove proteases. Proteases are genes that encode for proteins, and they're called proteases that chew down proteins. So if they're in the soup or in the fermenter, they start clipping away at the antibodies or the vaccines. And by the way, in that program with Zappy, when we removed 9 proteases at our 9X strain about 1.5 years or 2 ago, we ended up with this 300 times more productivity than the second place cell line And now we've knocked out 14 different proteases to make more stable, higher pure proteins at very, very low cost. And we're working in addition to a COVID-19 vaccine of our own and the variants and other opportunities in infectious disease, influenza, and other ones that I can't talk about. And as we have shown success in the ZAPI program and in our own internal and other third-party programs, we're gaining traction by people on a global basis to approach us to do fully funded projects on infectious disease and other drugs. And then, of course, we initially were working on just human therapeutics, but now we've been funded to work on growth factors. And growth factors are being funded fully by a company called Turtle Tree Scientific, where they're fully funding that program. And growth factors are used potentially in cell and gene therapy and other applications to rapidly multiply and replicate cells faster so that you can get these cells produced in larger quantities at lower cost. So it's really the same theme throughout biotech. The proteins are needed on a variety of uses and applications, either as an intermediate, like in a growth factor, to multiply cells using gene and cell therapy and other factors as well, or as a drug, as a vaccine at a more affordable, lower cost to bring a global population healthcare that heretofore they couldn't afford. And we're working on fully funded proof of concept expression programs from Big Pharma. So one of the things we're doing on our own, as I mentioned earlier, is we're developing through a proof of concept, like we engineered the C1 cell line to remove the proteases to generate a cell line now that we believe is the most productive, most powerful, fastest, quickest way to make recombinant subunit protein antigens or vaccines. We're working on now engineering the C1 cells to make monoclonal antibodies with the right characteristics, with the right glycosylation, with the right patterns and the right properties at the right levels. And we've been able to demonstrate hyperproductivity on our initial cell lines. And now with these stable cell lines, where the proteases are knocked out, we've been successful at doing glycoengineering. And now we got to put all three pieces of the puzzle back together again. And if we do that, we'll have hopefully one of, if not the most productive ways, the fastest ways to make monoclonal antibodies, which are about $200-300 billion in business today, and is the fastest growing segment of the pharmaceutical industry. So high value proteins, competitive advantages using C1 technology. So what is C1? C1 is a filamentous fungal cell that's been evolved and developed over the last 25 years for industrial biotech, and over the last 4 to 5 years being re-engineered, taking the power of industrial biotech and bringing it to an inefficient world that's used to making small amounts of expensive things in tanks, when we're used to making massive amounts of things in tanks very fast at very low cost. So competitive advantages is purity, productivity, robustness, speed, cost. I'm not going to get a lot of details here. I think you get the message that you can see from this slide that we can make record levels of proteins, whether it be an antibody in a short amount of time at higher yields at lower cost, or whether it be a COVID vaccine or other infectious disease vaccines like influenza, trispecifics. And so the growing numbers of types and classes of antibodies, therapeutic proteins, and vaccines that we're expressing in C1 is growing every quarter. So return on investment, how are we going to make money out of all this? Well, just like we did industrial biotech, we did non-exclusive license deals where we got upfront cash, then we're going to get access to milestones and royalties. And we generated over $30 million in upfront cash, non-dilutive money on the industrial side through those licenses. And then, of course, we can license small biotech companies that don't have the cash and get equity, milestones, and royalties. And what we're doing there is we're ending up with pieces of equity in these little companies that could grow up to be the next Genzyme or Amgen or Biogen or Genentech or Pfizer. And some of these companies, in the case of BDI in Spain, we own 16% of that company and used our technology as our equity stake. And if they develop products using C1, will end up getting milestones and royalties. And that's how we do it. Small guys and the medium size, they can't afford to spend their money in upfront cash. But this way we get more products into the clinic potentially that gets more milestones and royalties and gets an equity kicker as well. And then grants and contracts, for example, the ZAPI program, the funding we received there, and the Israeli government. And it wasn't just about the funding. We advanced our technology platform immensely based on their dime and their funding to us, in addition to all the work they did on all the animal studies, had no cost. And then, of course, we're developing things, as we mentioned, like Opdivo, our own COVID-19 vaccine candidate, heading towards the clinic that we're going to use as a proof of concept to show safety in human beings. And then we can reload it very quickly. And I've actually started to create the new cell lines. So when we have that data, we can rapidly go after the variants and make broader covering, deeper, lower cost, more affordable, larger quantities of variant COVID vaccines, and then the glycoengineering and other things we talked about. So investment highlights, many shots on goal, almost endless. It's kind of like the pearl business. Put a piece of sand in an oyster, out comes a white pearl. Put a different piece of sand in, you got a black pearl. And what we're doing is we're putting gene sequences in our cell line that are being given to us by people who have spent enormous amounts of money identifying drug candidates, vaccine candidates that can help make cancer a thing of the past, or rheumatoid arthritis, insulin, diabetes, etc., or vaccines for COVID-19, COVID-19 variants, or other pandemics, or other infectious disease. And then as a global business development, we're growing our footprint throughout Asia, India, Europe, and all over the world. Our board and our management team is stellar. They're motivated, they're driven. We don't have a lot of time, but I can point out Patrick Lucy, who just joined our board, was a 20-year veteran of Phoenix, and he was their chief business development officer. Phoenix got sold to Ligand for $500 million last year. And they had a similar technology, but it was based on a microbial E. coli bacterial system, which couldn't make 80% of the world's proteins because it can only make simple proteins and not complex, difficult proteins that are glycosylated. At C1, we can handle both sides of that equation, both the non-glyco and the glycoproteins. Barry Buckland, Merck, 29-year veteran, U.S. National Academy of Science engineer. Dr. Aaron Bose, again, U.S. National Science Academy engineer, 34 years at Pfizer. Michael Tarnock, our other board members are very strong as well in our Audit Committee and corporate governance. So we're satisfying unmet needs that the world has to make healthcare accessible and affordable on a global basis, not just in a pandemic, but in animal health and human health for a variety and a number of growing opportunities. And our aims and milestones are to really dominate and disrupt the biomanufacturing process, just like Elon Musk has disrupted the car business with the electric cars. Our vision is to accelerate the adoption and use of our technology broadly and widely so that it becomes in all the scientific labs and all the corporations, and it replaces for at least some, if not most, of the drugs and vaccines that need to be made in large quantities and more affordably. And again, our expanded infectious disease franchise based on the success we had in ZAPI with the Schmallenberg virus, Rift Valley fever virus, and now with COVID-19, we're being approached by multiple parties to expand on that as well. And then again, as we talk about it, we have a global basis, a global reach, expanding footprint. And now I'll take, I think, time for calls or chats.
Yes. Great presentation, Mark. You questions. So starting with the first one, variants are threatening the efficacy of the vaccines. What can your technology do to combat variants now and in the future?
Yeah, so it's interesting because we showed a slide here. What we can do with existing things like the South African, the UK variant, or the British variant, or the California variant, the New York variant. I now hear there's a Japanese variant. There's a Indian variant. So the beautiful thing here is we can reprogram these cells very quickly. Within 7 or 8 weeks, We can take out the Wuhan gene, put in the South African gene, and have a stable cell line. So once we get our proof of concept safety in humans, we can then just swap out the different gene sequences to make the different variants. And what makes us unique and novel is not only we can do it quick, the cell lines, like the Wuhan cell line we have, we can make billions of doses on a global basis using standard microbial fermenters that use E. coli. And they're on virtually every continent and in many countries all over the world. So that's how we can deal with that. And we can make them in the quantities that allow you to make a tetravalent or a trivalent vaccine, which is either 3 or 4 of the variants in the same shot. Just like the seasonal flu, you have 4 different proteins. So if people are struggling to make one protein in the quantities you need, how are they going to make 3 or 4? And I don't think in the case of mRNA, you're going to have 3 or 4 genes in a cell line that gets put in your body and asking your body to make those 3 or 4 genes in the right ratio. But we'll see. So I think we may have really the most viable, realistic approach. And as the VP of Science and Development at Meditox said, he tested CHO cells, he's tested insect cells, which again are being baculovirus used by Novavax and Sanofi, and they switched to C1 cells, and he said it's the most realistic approach for the COVID vaccine variants.
Fantastic. Can this technology also be applied to the flu? What other diseases does it combat?
Yeah, actually, we did a project in 2015 with Sanofi Pasteur, and we demonstrated that we can make hemagglutinin, which is one of the components of, or actually 2 of the components of the 4 components from a seasonal flu. And we demonstrated that, actually they demonstrated, it's their data. You can find it on Dyadic.com, Media Center presentations, and you can scroll all the way down. And they demonstrated that with 10 microgram dose in the mice trial, we had 100% protection versus 50%. And so when you triple that to 30 micrograms dose in the mice, they got up to 75%. Of course, we were still at 100%. So we could use 1/3 the amount, get a better result, and make it 50 times cheaper. And we're working with Oslo University right now as well on hemagglutinin and on the flu vaccine. So any infectious disease, if it's an antigen and it's a glycoprotein, I think we have the system of choice to not only make it where it's safe, effective, and protective, to make large amounts of it at flexible commercial scales more affordably.
So tell us what is proprietary about your technology that the majors in the space don't just have yet.
Well, the majors in the space continue to just do the same thing over and over again, expecting different results. And of course, Einstein defined that as insanity. And that's why we are where we are today. They didn't care about infectious disease in a significant way until COVID showed up. It's not the most highly profitable part of the business. But when it comes to recombinant protein vaccines, and I'll highlight for you, in 2019, if you go online and you type in President Trump, executive order, influenza vaccines, they pointed out there was a shortfall in the manufacturing technologies to make vaccines for a pandemic. And so he points out 2 directions: cell-based vaccines and cell-based cell lines and expression systems. That's like mRNA, like Moderna and Pfizer. And they talked about recombinant cell lines and expression systems, which is exactly what we have. And we didn't have to create it when the pandemic hit. We had it. We spent 25 years creating it on the industrial side. We spent 5 years with the Zoonosis Anticipation Preparative Initiative. And on our website, in addition to the 2 videos that I tried to play today, and you heard one, they said if nothing else came out of that 5 years, C1's fantastic. And there's a 8-hour presentation of a 5-year review on our website at Dyadic.com, Media Center presentations on February 4th. And you can watch to your heart's content of, I think it's 14 different presentations reviewing 5 years of work. And C1 is demonstrated in there throughout as the most productive and key role in making the quantities and the qualities and at the cost the world needs.
Thank you for that. So the White House is reviewing if they should lift intellectual property shields on COVID vaccines. Will this put Dyadic in jeopardy?
No, because first of all, we'd be happy to provide our cell line, which we have done with the Israelis, the Europeans, the Koreans, and others. What we're doing is, again, although we have our own vaccine candidate going forward, and as we talked about, we're going to prove in human beings it's safe. We've already demonstrated it's safe in cattle, it's mice, and sheep. We have US FDA GRAS status, which means generally recognized as safe on the cell line for food and feed, where we had toxicology and pathogenicity. But we hope, in fact, that people come to us and give us their gene sequences. And by the way, several people are, and we're talking to others, because we're willing to make vaccines using our technology for everyone that has a potential vaccine that can bring safety and protection to human all over the globe. So that would be actually not a problem for us at all.
Good to know. Have you been approached by any of the majors yet?
Well, we're in discussion with the majors, and actually the majors are funding us for several different products before COVID and during COVID, not for COVID itself, but we are in discussions with some of the majors to talk about how with these variants we can change the game. So in some ways, we believe with the COVID-19 variants, we're even in better shape to help the world than Big Pharma than we were last year.
Fantastic. So what's your current burn rate per month? And do you plan to do another capital raise?
Yeah, so it's a good point. So we have $29.2 million in the bank, no warrants, no debt. If you look at last year, we gave projections of $8 to $10 million. I think we came in under $7 million in cash. This year, we gave a projection. Actually, our conference call was last night of 10 to 12 million. It's a little higher than it was last year and a little higher than the year before. And the reason for that is we're going into this first phase 1 clinical trial, but we're not going to go and move into phase 2 on our own dime. We're looking to partner that out on the different multivariants, tetravalent, trivalent, quadrivalent vaccines. So we're not going to be spending our money to go forward into phase 2. We're spending our money to prove safety and preliminary efficacy, not just for COVID-19, but when we do that, it shows for other recombinant proteins and vaccines that they can be made safely using our technology. So that's going to hopefully accelerate the opportunities. We're already immersed in more than we can probably handle, but that's going to open up the floodgates even further.
Fantastic. So talk to me about some of the major biotechs and pharmaceutical companies in this space. Who would your technology marry well with?
Well, I think our technology marries well with any and all of them. The question is, will they use what they have and fall short and continue to get 1 billion doses out for a 16 million dose world at too high of a cost? Or do they want to keep it simple, stupid method, which is load a program, gene cell, into a cell line that's more productive, that can be grown in standard microbial fermenters. So you don't have to buy fancy equipment. You can use E. coli fermenters that are available in virtually every region of the world, and where we can produce not only in massive quantities in big tanks, but as the Dr. Brick said, the video you didn't get to hear, but that you can shrink down the footprint or use smaller fermenters and still pump up a lot. So to give you an example, there's a company, CGMP, like the Wuxis or the Catalents or the Pathians or the Lonzas. Lonza has 2 15,000-liter fermenters in Europe. And within 5 days, we believe at 3 grams per liter, at 70% recovery rate, they can produce enough antigens for a single, like a Wuhan antigen, in a week. And that doesn't mean they can put the vaccine together in a week, but the active ingredient. And that's a number that's unbelievable. So Big Pharma needs to take notice and to really look at doing something other than what's invented here and to apply a technology platform that can help the world achieve and get us all back to some sense of normality.
Amen to that. So now that the industrial non-compete agreement has expired, do you guys have plans to enter the industrial area again?
Well, we're being approached by a number of people, and we're looking at it and we're evaluating what the best way to do that would be and if we have the desire to do so. But I could tell you that it's a good possibility because, again, it's a platform technology that makes proteins. Proteins are enzymes, proteins are antigens, proteins are antibodies, proteins are therapeutic drugs, proteins are growth factors. I can go on and on and on. So what we're doing and what people have to realize is we're not taking on all this responsibility. We're doing the same thing over and over again. You're giving me a gene, I'm turning it into a protein by putting it into a cell that can produce more for less at flexible commercial scales. So that's what we're doing. We're programming cells for the people on the planet that have uses and applications that are growing and emerging. So we're not spread too thin. And the beautiful thing is we're working with VTT in Finland. And VTT is a Finnish government research group of 2,200 scientists, 150 scientists in the biotech group. They spent over 3 decades, like we have, engineering these types of fungal cells. And we have a long-term agreement, and they're some of the best people in the world to engineer these cells. So we've been able to ramp up when we have new projects and expand them using them. We also have BDI Pharma, which we own 16%. They're the former scientists from Abengoa Bioenergy. They've spent working with C1, I think now since 2007, so 14 years. So we have engineers that know how to program these cells and how to grow these cells and how to scale these to levels that nobody's going to need in the pharmaceutical industry. Because you remember in the biotech industrial business, we scaled up to 500,000 liters, the enzymes they use for biofuels.
Mark, as always, great presentation. Thank you so much for all of your wisdom in this field. How would you like to close? Close maybe telling us where we can find you, your website, and any closing remarks you have to end this presentation.
Great, thank you. Well, you can find us all over the globe because we're working with partners on a global basis. You can find me, he's in Jupiter, Florida. It used to be on an airplane everywhere. So once we get out of the COVID world, which we're helping to do quicker and sooner, more affordably, you'll find me at different conferences, making different presentations. You'll find our scientists making scientific presentations. But where people can find out about the company is www.dyadic.com. Go to our media center, look at the presentations. I think you'll be shocked at some of the scientific presentations and statements coming from very conservative scientists that are basically saying, we're here at the right time, at the right place in history to make a difference. And that's our goal, is to bring human health and animal health, not just COVID-19, but traditional pharmaceutical products that people are not getting access to, and disrupting this space in a way that's positive, constructive, and profitable for humankind and Dyadic shareholders. And thank you for having me.
Fascinating. Thank you so much for all that you guys are doing. Really looking forward to your updates and progress.
All right. Thanks for having us, and stay safe, everybody. We're working hard to keep it that way.
Thank you, Mark. Appreciate you.
Great. Thank you.
All right, everyone. We are going to take that quick little minute break so we transition to the next presenter. There's going to be a black screen. Don't go anywhere. Stay with us. And if you're on that Apple device, click the play button to launch the next live presentation. We'll be right back.